will be talking about norovirus. The norovirus is a very small but deadly virus that is responsible for symptoms such as explosive diarrhea, vomiting, fever, and also gastroenteritis, which is inflammation of the intestines. The norovirus is annoyingly successful because it can be transmitted very quickly and shed very quickly without presenting any symptoms. By the time that symptoms have been presented, it is possible that the infected person has infected many people already. Additionally, norovirus is a non-enveloped virus, which looks like this, as opposed to this. This means that a lot of the disinfectants we have, which act on the lipid envelope, will not be applicable for the norovirus, so it is resistant, and it is also resistant to drying out and heat. This means it is very stable on surfaces. In fact, it has been shown that noroviruses can survive for up to 3 years in wastewater. Therefore, you must wash your hands with warm soap and water when handling food and identify sources as early as possible. And where are the sources? Well, because it is shed quickly and because it can be transmitted quickly, you often get norovirus spreading in small confined spaces, well, not necessarily small, but confined spaces where there's lots of crowding, such as on cruise ships, childcare centers, and nursing homes. This small but deadly virus attacks everyone. In terms of diagnosis, you can take a swab either from the person's feces, from the surface, or from the food. And then you can use reverse transcriptase polymerase chain reaction or electron microscopy to analyze the genome and determine, you can use electron microscopy to view it and then see whether if it is the virus, but to be more precise, you can use electron microscopy. So what does the current research tell us? The current research has determined that it is a positive single-stranded RNA virus which belongs to the Calliciviridae family. The genotype G2.4 is responsible for most of the norovirus infections we see, and this is just an example of its genome. And in terms of replication, it is in several stages. So the whole genome is replicated once, and then it is broken up and then replicated in several stages. This allows it to coordinate when certain proteins are made, making sure that enzymes are made first before capsid proteins are later on made. As you can see, so you would encode for a long strand first, where the non-structural genes are on this side, so the enzymes, and then you would code for the structural genes later, which makes sense because you need enzymes to code for the structure. So these are all of the proteins that are known in the norovirus genome. You have the NS proteins 1 to 7, each coding for different enzymes. Then you have VP1, which codes for the capsid. And then you have VP2, which just codes for proteins within this virus. Let's look at some of the proteins that they use. So there's NTPase, which provides energy. And this is made by a gene code NS3 and NS4. You have VPG, which is a viral protein genome, which is a genome-linked viral protein that is attached to the 5' end of the genome usually, and this just facilitates RNA synthesis. You have something called a protease, which just helps to break up this polyprotein chain, which will be encoded by this RNA into smaller individual proteins. And then you have something called RDRP, which is RNA-dependent reverse polymerase. RNA-dependent RNA polymerase, which just catalyzes the replication of RNA from the RNA template. In terms of other sciencey stuff we know about this norovirus, it prefers to attack dendritic cells, monocytes, and macrophages. There are currently no small animal models to study the norovirus, but there has been some research in zebrafish and also mice. And these studies have shown us that if you have something called the VCD300 IF receptor, you are more susceptible to infection. If you're immunocompromised, you can also become more susceptible to the infection, especially if you are lacking in STAT1 or RAG2 genes. And that was shown in the mice. So we assume that the same thing would also happen in humans. If you have a healthy microbiota, this is a good thing generally, but in terms of the viral infection, this can be not so good because healthy microbiotas help to downregulate our inflammatory response, especially interferon gamma-driven responses, which are helpful in defending against the norovirus. So in conclusion, the norovirus is a virus which has managed to maximize its survival potential by making the most out of the healthy microbiota that we have. It is a virus which can transmit very easily and can survive for services very long, and it has adapted its structure to do so. And if you want to stay safe, remember to wash your hands with warm soap and report cases of food poisoning so that they don't spread to other people. Okay, see you!